All right. Hello everyone and welcome to Quantized Bio with Dr. Casco. In today's video, we're going to continue chapter 11. We're going into section 3, signal transduction. Cascades of molecular interactions transmit signals from receptors to relay molecules in the cell. Okay, in the last class, we talked about signal reception, where we saw how ligands can come into the receptors that are on the cell membrane to initiate a relay of molecular transduction, ultimately for the cell to get a response or to elicit a response in the cell. So we're going to go into that second process called signal transduction. Okay. Usually, this transduction stage that you see here is a multi-step pathway, all with the purpose of amplifying the signal. And this involves an activation of proteins, which usually in itself involves the addition and removal of phosphate groups on the particular protein. We're going to see that in a bit. And also, it could be a regulation of small molecules and ions. We're going to see in particular cyclic AMP or calcium ions. So let's get to it. The benefits of having a multi-step transduction system is, even though if you have a initial ligand on the receptor will then elicit a response from one molecule or just a small amount of molecules that you see here. I'm going to zoom in a little bit. This multi-step process means that one molecule can then activate another layer of molecules. In this particular case, you have this first amplification where you have enzyme number 1, let's say, and you have 3 of them. And then each of these enzymes, enzyme number 1, can then amplify and activate, their own set of 3 molecules, and then more and more and more. So this idea of having a multi-step process can help the cell coordinate better, can amplify a signal a lot faster, ultimately for the cell to elicit some type of response from the stimuli that it is obtaining. Okay, so signal transduction pathways, getting more specific what is happening now. This is the multi-step, and this overall layout is generally what happens. Let's get into more detail now. Okay, so again, like I mentioned before, it begins with the binding of a ligand to a receptor on the plasma membrane that will elicit a signal transduction, ultimately a chain of molecular interactions, so you can have a cellular response. You can think about this whole signal transduction pathways as a domino effect, okay, which often involves proteins. And this protein interaction usually involves the interaction between protein number 1 to protein number 2. Okay, so this in itself is called protein-protein interactions, fundamental to cell signaling. Okay, usually, most often than not, the original signal molecule doesn't pass, or even the ligand doesn't really pass along the pathway or even enter the cell. So the signal, hence the name transduction, right? It's transduced, it's passed through from relay from one person to the other, one molecule to the other, to the other, to the other, ultimately to get some effect. Okay, usually that relaying of signal is through conformational change, meaning that some proteins will change in their form in some specific way. Okay, and it could be induced by phosphorylation. We've talked about phosphorylation before, right? So protein phosphorylation and dephosphorylation. So you have a conformational change. Whenever you attach a phosphate group to a protein, it will change the shape, activate it, okay? And usually this is done by what are called protein kinases. These protein kinases enable the phosphorylation of some other enzyme, including other kinases. Okay, so big thing to note here, protein kinases. These are a group of proteins, a group of enzymes that can enable the phosphorylation of some other thing. It could be another kinase, it could be another protein that you need, okay? But what you need to understand is protein kinase, okay? So these protein kinases, they can act on other proteins, as I mentioned before. They could be substrates, they could be other kinases, and so forth. They act particularly on specific amino acids like serine and threonine. Serine, threonine amino acids of the substrate protein. And then they can actually act in sequence where they activate one kinase and then another, kinase number 3, number 4, and so forth. This is usually called a phosphorylation cascade, like a water cascade, right? Where everything's flowing down in sequence, one to another to another. Okay? And ultimately, you want to have the activation of a protein to elicit a cellular response. So here's a visual of what I'm talking about. You have a signaling molecule. That signaling molecule will activate a receptor. Receptor will change shape, activate some relay molecule, which will in turn activate a kinase, a protein kinase, which will become the active form. This protein kinase will help phosphorylate protein kinase number 2, which in turn gets activated. And then protein kinase number 2, depending on the pathway, you can have multiple kinases. Okay. In this case, you just have 2 kinases that will activate the actual protein, which will then promote that cellular response that you need. Could be gene activation, cell growth, division, whatnot. Okay, depending on what we're going to see here. Okay, kinases is important to note. All right, let's continue on here. So around 2% of our genes codes for protein kinases, which is quite interesting. And there's a good variety of protein kinases in our body, depending on what you need. But because there's a lot of variation, this is in particular of particular importance in medical science and in medicine, because there's many disorders and diseases that are involved with mutations to these kinases that could lead to abnormal cell growth, cancer, and tumor development. So scientists monitor these type of proteins and to figure out what is the root cause of certain diseases there. Okay. I'm just gonna be a little bit redundant and just say again, whenever you phosphorylate a particular molecule, that will essentially activate the molecule or the protein. It will become from an inactive form to an active form. Okay. How about when you want to turn off these kinases? It is not good that they're on all the time if you don't need it. Okay, you're going to waste ATP. You can over-induce a cellular response, which can also have detrimental effects. So there's another set of proteins that you need to know that are the phosphatases. Protein phosphatases. These are enzymes that will help remove the protein, the phosphate groups, okay, in a process called dephosphorylation. So you have kinases that will phosphorylate, and you have phosphatases that will dephosphorylate, that will take away the phosphate group. Ultimately, what's going to happen is that the signal transduction will turn off. You are turning off the pathway whenever you are removing phosphate groups, okay? And this is important because if you turn off the proteins, that could enable a signal that will come again to, restart or jumpstart the pathway again if you need it. So again, it helps to regulate the signaling system, okay? So again, the the whole idea of signal transduction is a balance. It's a play between active kinase molecules and active phosphate molecules. And depending on the concentration difference, you will have a signaling passing or not. Okay. So that's important to know, right? There's a balance between active kinase molecules and active phosphatase molecules. All right. So just to kind of highlight, what these sentences in yellow is very important to note, okay, for your classes and to understand these particular things. When the extracellular signal molecule is absent, you will have active phosphatase molecules predominating in the cell. Okay, of course, and the signal pathway will shut down. All right. Sorry about that. And then continuing on to the passing down the signal, we have another set of messengers that in the biology textbooks we generally call second messengers. Okay, and there's a couple of types of second messengers, and these could be small water-soluble molecules. They're non-protein molecules. They're referred to as second messengers. And amongst these second messengers, as I mentioned earlier in the video, you have cyclic AMP, okay, or CAMP, or calcium, +2 ions, which I'm going to just call calcium ions, right? In the signal transduction pathway, the first messenger is categorized as the ligand. A ligand is your first messenger that will transduce and elicit this signal transduction pathway. Second messengers are then diffused throughout the cell, and they will pass, the message along. And they're usually initiated through these G protein receptors or these tyrosine kinase receptors also. Okay? And depending on their concentration, again, concentration is very important. You will elicit the signal pathway. So they're very, very sensitive. It is important for the cell to have a tightly regulated concentration of these secondary messengers, or these second messengers. Okay, if you remember in the previous video, we're talking about epinephrine and how epinephrine elicits a signal transduction pathway to ultimately take glucose out from glycogen to burn. Okay, we're going to continue with this example here. Binding by epinephrine leads to an increase in cytosolic concentration. So concentration in the cytosol of cyclic AMP. Okay, and just to go into the next little visual here. All right, so whenever you have this kind of activation from epinephrine, being the ligand into a receptor, what happens is that you get an activation of a particular enzyme called adenylyl. Sorry about the pronunciation there. Cyclase. Okay, and this is an enzyme that will convert ATP into cyclic AMP. Okay, CAMP in response to epinephrine. So every time you have an epinephrine signal, this will boost, okay, cyclic AMP 20-fold within seconds. Okay. After you have used, or a cyclic AMP has done its thing, you have another enzyme called phosphodiesterase that will turn off and break, break these bonds within cyclic AMP. Let me just zoom in here. Ultimately to produce adenosine monophosphate, okay, a precursor to ATP. All right, so what is happening? Going back into the signaling part of this story here. You need to have an active ligand, a signal of epinephrine, to increase the concentration of cyclic AMP. Many hormones and other signals trigger the formation of this compound, okay, of CAMP. I want to call it CAMP. It's because it's so important. It's tightly regulated. Many signaling pathways use cyclic AMP. Okay. G protein receptors, RTKs, and protein kinases are involved in these cyclic AMPs. So it's so fundamental to signal transduction. For this particular molecule, we have a specific protein kinase A that it is involved with. Okay, so C-CAMP, cyclic AMP, diffuses through the cell and it activates, again, a serine threonine kinase that is in particular called protein kinase A. Okay, protein kinase A. And then this kinase will phosphorylate. Remember, it's a kinase, so it's gonna phosphorylate other proteins depending on the cell type. I'm gonna get back to that later on. Even so, it's so important, protein kinase A and cyclic AMP is so important that even microbes, right, bacterial infections and can cause disease that disrupt this whole relay of cyclic AMP and the whole G protein signaling processes involving cyclic AMP. An example of this is cholera bacteria, which, Vibrio cholerae, which you may know or not know. It's contaminated, right? It's present in contaminated water with human feces. And if you're drinking contaminated water or ingesting something that is contaminated with this bacteria, you will get diarrhea. Okay. This bacteria in particular, it colonizes the small intestine, makes biofilm, if you remember from the previous video, and it will produce a toxin that will change and alter this G protein and ultimately produce an effect in terms of salt and water secretion. Why is that a problem in the intestine? This will induce diarrhea and it ultimately make you use the bathroom, severely. So it's like if you have an active laxative in your body, hence the diarrhea there. Okay. Back in old days, you can even die from this. Nowadays, of course, we can treat this relatively well with electrolytes and, staying hydrated. Okay. So, yes, more specifically, just to kind of finish off the notes here regarding this bacterium, the modified G protein is unable to hydrolyze GGP to GDP and remains stuck in the active form. So it's continuously creating cyclic AMP. More cyclic AMP means that you're going to secrete lots of water and salt into the intestines. Okay? So very important there. Another pathway uses cyclic GMP, very similar precursor molecule, as a signaling method. And in this case, you have smooth muscles in arterial walls. Okay, this will induce cyclic GMP will induce the relaxation of smooth muscles in the arterial walls. It's quite interesting that scientists developed a compound very similar to this, and it was used to treat chest pain. But this compound inhibits the hydrolysis of cyclic GMP to GMP. So what happened? You have the signal activated for a longer period of time, but because this is a medicine, it was going throughout the whole body, and then scientists figured out that this compound also activated blood flow to the penis, to the male reproductive organ, which turned out to be Viagra, which I'm sure a lot of us, just out of diffusion in media, what this medicine is or have heard of this medicine. Okay, so you have Viagra, which treats erectile dysfunction because this molecule will activate a dilation of blood vessels, therefore more blood flow in that particular area. Okay, just in essence, to give you a sense as a visual here, what is happening with cyclic AMP. Okay, you have your G protein receptor, you have the first messenger, in this case, let's say epinephrine, comes in, activates the receptor will activate the G protein, will remove GDP and put GTP. You guys remember the visual in the previous video, it just said enzyme, but now we're getting more specifically adenylyl cyclase. This enzyme gets activated, protein kinase A, okay? Which in turn will activate cyclic AMP, sorry about that, which will then activate protein kinase A, ultimately eliciting a cellular response there. Okay, so cyclic AMP, very, very important. Another important molecule, second messenger, is calcium ions and inositol triphosphate. What is this? Let's talk about it. So these are signaling molecules, right? Many signaling molecules are needed within mammalian systems to carry out neurotransmission, growth factors, hormone regulation, and so forth. All these processes involve a certain concentration of calcium ions. Okay? And to induce responses in their target cells via transduction pathways that increase cytosolic concentration of calcium. Sorry, so this signaling will induce a concentration increase or some regulation of calcium concentration. Okay. Okay. So what happens? In this case, this idea of concentration is very important. All right. Calcium is even more widely used in cyclic AMP as a second messenger. Okay. In animal cells, you tend to have, or an increase in calcium concentration can cause several things. Contraction of muscle cells, secretion of vesicles, of substances, okay, generally by exocytosis, cellular division, greening of plants, okay, in response to sunlight, okay. So in plant cells, you have an increase of calcium ions. It can trigger a response, okay, such as greening, production of chlorophyll or whatnot within the, the chloroplast in response to light. So cells use calcium as a second messenger to, within G protein pathways and RTK pathways, ultimately to create a response. What is important to note is the concentration difference. Okay, generally within the cell, calcium ion concentration is higher outside of the cell or within the ER. In the cytosol, you tend to have very low concentration. Okay, so this is essentially what this picture is showing. And the cell uses ATP to try to keep that concentration in the cytoplasm, in the cytosol, relatively low. Active transport and whatnot, you're pumping calcium into the ER, into mitochondria, even outside of the cell, whatnot. Okay? What's important to note is that calcium concentration is generally lower inside the cell. Okay? By quite a lot. This is probably overkill of information. By a factor of 10,000 or more, a good amount. Okay? Proteins actively transport calcium outside of the cell. Okay? So you want to keep a good concentration difference. Okay? Now, the reason for that, so the cytosolic calcium is low, so small changes in calcium ions can cause a relatively large percentage change in calcium concentration. Why is this important? Because, right, whenever you release calcium from the ER or the mitochondria, or bring it in from the extracellular environment, okay, This, activation of calcium can trigger cellular responses. Okay, now how does this actually happen? Calcium release, it involves some other messengers, okay, diacylglycerol and inositol triphosphate. So these two can activate calcium release. Okay, these two messengers activate calcium release. Okay, and then these two are created by what is called phospholipase, comes in and cleaves some phospholipid molecule on the membrane. So I'm going to show you the visual really quick. Just going to go over the notes. Okay. So phospholipase in itself can be activated by a G protein or an RTK, right? A tyrosine kinase receptor. This inositol triphosphate is referred to as IP3. IP3 activates the calcium channels to open, releasing calcium from the ER into the cytosolic environment. Okay, and then when calcium comes in, it can elicit a cellular response. Let's take a look at the visual here so you can get a better understanding. You have a G protein coupled receptor followed by some, ligand coming in that will activate this receptor. This receptor, of course, you guys know, will activate a G protein. This G protein will get that GTP onto itself, activate an enzyme. In this particular case, you have phospholipase C. This phospholipase C will then cleave, will then break some compound. In this case, you have PIP2, okay, which has this little submolecule. Ultimately, this compound will create inositol triphosphate and this DAG molecule. And these molecules can come in, and then they themselves will then attach to a calcium gated, an IP3 gated calcium channel. So this particular molecule is specific to this protein, okay, this calcium gated channel. Open it, which will enable an influx of calcium to come into the cytosol. And then ultimately, proteins can be activated, and then you get a cellular response there. Okay, so that's what's happening here in terms of calcium as a secondary, sorry about that for the microphone, as a second messenger, and also cyclic AMP. So make sure you understand that particular idea there. Okay, we're going to move on to the next, to the last part of the signal transduction pathway, which is cellular response. Cell signaling leads to regulation of transcription of cytoplasmic activities. Okay, and you can have responses occurring. You already got the signal transduction, ligand, receptor, relayed molecules, and protein kinases, ultimately to create the secondary messengers to go where it needs to go. Where can it go? It can go into the cytoplasm or into the nucleus. So you can have responses, nuclear responses or cytoplasmic responses. Okay? And ultimately, either which way you go, you can have protein synthesis involving DNA and turning on and off of particular genes. Okay? You can activate a steroid receptor, which can activate a transcription factor. And transcription factors, they're molecules that can regulate several genes, different genes. Okay, this is very important. Depending on the signal pathway that you use, right, you can regulate the activity of proteins rather than their synthesis. And then this in turn can affect proteins that function outside of the nucleus. So there's a cascade of regulation there. And I'm going to get into that in a bit here. Okay, let me just zoom in so you guys can see the. Actually, let me move this here so you guys can see better the notes here. All right. Okay, so signal pathways can regulate, as I mentioned before. And a signal can cause, again, the opening and closing of an ion channel or a change in the cell's metabolism, depending on what the effect is. Are you going into the genes? Are you turning on some or turning off some genes? Okay, what exactly is happening? It depends on the signal pathway. In the case of epinephrine, all the way from the cell membrane, relays a signal into the cell, right? Which can regulate cellular energy metabolism. Ultimately, we have that end goal there, right? You want to break down glycogen so we can have a fuel system, so we can be burning energy in this whole thing of flight or fight response. Okay? Epinephrine can release millions of glucose molecules in a short period of time. Nevertheless, you have this idea that the signaling events of whatever signal pathway comes in can elicit different things. Nuclear responses, cytoplasmic responses, cell division, okay, activation of genes, repair, and whatnot. This is critically important, again, in medicine because amongst these is cell division. And then when you have abnormal cell division, usually that leads to cancer. Okay? Usually that leads to cancer. Now, before I get into the idea of amplification, I just want to, show you the visual of what I'm talking about. Okay? So you have some ligand activating a receptor, and this ligand, maybe it could be a growth factor. Okay? Fibroblast growth factor, FGF maybe, which will elucidate this whole phosphorylation cascade. I won't go into so much detail there. You can see in the little visual. Ultimately, that last protein that gets activated can migrate into the nucleus, okay, which can activate special proteins, special molecules that are called transcription factors that will help turn on some gene to activate transcription and then translation. Yeah. Very, very interesting stuff here. All right. And then I want to kind of go into this idea of amplification. Remember when we talked about the multi-step process, we're going to go back on this. Okay. Signaling pathways, as I mentioned before, they have multi-steps. And this idea of multi-step can help amplification. Amplification can help quick response, very fine-tuning response and coordination amongst the cell. Okay, and it doesn't how the amplification occurs does depend on the nucleus or the cytoplasm at particular points, but ultimately the end goal is the same. Okay, when you want to fine-tune, you want to coordinate and regulate the signal transduction event, you need to consider four things, right? Amplification of the initial signaling event, single signaling event, the multi-step cascade in itself, having a couple of control points, okay? And then once you have this kind of activation of relay molecules, your response is further enhanced by what are called scaffolding proteins, and I'm going to talk about that in a bit. And then ultimately, you want to figure out how to terminate the signal. You don't want to have the signal all the time, right? So signal amplification, we know already in the case of epinephrine, it triggered the cascade. It produces the products of each subsequent step are greater than the preceding. So you have an amplification effect. I'm going to zoom out just so you can see what is happening there. So you have epinephrine, one molecule, and just that molecule activating that G protein, you have an increase in the amount of molecules that can be creating. Okay? And then these G proteins in themselves are activating adenylyl cyclase, which then all of these adenylyl cyclase are activating cyclic AMP via a phosphorylation. And you notice 10 to the power of 4, 10 to the power of 2, there's many more molecules being amplified. Cyclic AMP then activates active protein kinase A, okay, which will then activate phosphorylase kinase, and then furthermore activates, other compounds. Sorry about that. Other proteins. Ultimately, to elicit that cellular response where you have glycogen being broke down to glucose 1 phosphate. And then you have 10 to the 8 molecules. Just with one molecule of epinephrine. That is quite mind-blowing. Okay? So again, yes, there is regulation amongst this cascade here. So the amplification effect, the proteins persist in their active form for long enough to process multiple molecules of substrate. Okay, so you want, of course, to be there long enough to be able to activate many molecules. Okay. So specificity of cell signaling and coordination of the response. This is very interesting here. Okay. Now imagine, think about this idea about having epinephrine. And epinephrine can have different effects depending on different cell types. Why is that? If supposedly we have all the same genome and genes in the same cells. The reason being, different cells actually activate different things, different genes, so they have different proteins. Just to give you a sense here, there's 2 cell types. Let's say we have a liver cell and a heart muscle cell. Both are in contact with blood. Epinephrine flows through the blood, okay? But signals can trigger different things within the cell, okay? Within each of these particular cells, okay? So in the case of the liver, you have glycogen breakdown, and in the case of heart cells, you have an increase in heart rate, okay? So this can elicit depending on the proteins that you have on and the proteins that you've made and the genes that you're turning on. Okay? So different genes can be used, different collection of proteins, different signaling cascades. It all depends on what you have in the protein, in the cell. Okay? The next thing, the efficiency of signaling depends on scaffolding. Now, before I get into the scaffolding, I want to just go over specificity of cell signaling. Okay? Just to give you a visual now of what I mentioned before. Same signal molecule if you see here. Now, depending on the cell type, here we have 4 different types of cells, cell A, B, C, and D. Okay, and I'm going to zoom out so you can see what exactly is happening there. Hopefully on YouTube, the screen's not small enough. The screen's big enough that you can actually see it. Well, so different cells can elicit different responses just because of the different proteins that you have can create different cascades. In this case, cell A, you have this, very basic one-way, relay of signal to make a particular response. In another cell, the signaling pathway can actually branch into multiple parts where this RTK can activate a particular protein, and that protein itself can activate 2 different types of proteins, which can create 2 different responses, response 2 and response 3. Cell C can activate response number 4, where you have a signal, some ligand A and ligand B working together, okay, in regulation, right, to create another response, and then another response, right, for cell D. So it all depends on the signaling pathway and the components of the signaling, that transduction pathway. It's incredibly it's fascinating how this actually works. And we're going to see some more examples there. Okay, So signaling efficiency, right? You have what is called scaffolding. Scaffolding is quite interesting in signaling complexes where you have, rather than relying on the diffusion of large relay molecules, many pathways have what are called scaffolding proteins. And these are large complexes um, of proteins that are kind of clumped together so you can have a kind of a set response. Uh, so these scaffolding proteins, they could be large relay proteins, and they can have, again, as I mentioned before, a complex where you have your scaffolding protein, and it can be carrying several kinases at once. They're in close vicinity, and if they're in close vicinity, you can enable them to quickly activate, so you can relay the signal pretty fast. In this particular example, if you see here, hopefully the visual is well enough, you can see the notes and the picture. If you're holding 3 protein kinases, they will, appropriately quickly activate from the, ligand receptor signal, okay? So ultimately, you can facilitate a phosphorylation cascade, okay? Some scaffolding proteins in brain cells permanently hold together networks of signaling pathway proteins at synapses. Okay? Again, always to elicit something quick. The book gives you an example of an inherited disorder. The Wiskott-Aldrich syndrome is caused by the absence of a signal relay protein. Some of the symptoms include abnormal bleeding, eczema, predisposition to infection, and leukemia. Just because there's an absence of this protein in immune system cells. Okay, and this WAS protein is located just beneath the cell surface, where it interacts with the microfilaments of the cytoskeleton and with several other signaling pathways, including those that regulate the immune cell proliferation. Okay? When the WAS protein is absent, the cytoskeleton is not properly organized and signaling pathways are disrupted. Okay, so just to kind of briefly read on this particular thing. Depending if you're an AP or an undergrad, you might need to know the specifics of that, but I just wanted to give that to you guys. Okay, so continuing on, so for a cell to receive new signals, each molecule change in its signaling pathway must last a short time. You don't, again, as I mentioned before, you don't want to have a signal activated so long. Okay? You immediately use it, you turn it off. If a signaling pathway component becomes locked into one state, you will have improper function beginning. Okay? As we know with processes in terms of enzymes, this idea of binding of signal molecules to receptors, they're usually reversible. Okay, in the sense that receptors need to return to their inactive state once the signal is broken down or released. You don't want to have a signal constantly active. Okay, some toxins are pretty much like that. Okay, and then of course, as you know, the signaling molecules, the ligands, concentration decreases, there are fewer receptors, and you start to have a play between active forms and inactive forms of the particular proteins, of these receptors there. Okay? Usually, in order to elicit a signal transduction pathway, you need to have a certain threshold reached of concentration of active receptors. Okay? When you don't have that threshold, then ultimately that cellular response will go down. Okay? So, you have all these molecules going back to their inactive forms. G proteins will ultimately go back to having a GTP, will hydrolyze the GTP to have GMP, okay, or GDP, sorry. Phosphodiesterates will convert cyclic AMP to AMP. Protein phosphatases will inactivate phosphorylated kinases, so they'll take away the phosphates and so forth. Ultimately, everything goes back to that kind of resting state for the cell to receive a whole signal cascade again. Okay, and then the last section for this chapter is apoptosis integrates multiple cell signaling pathways. Okay, so when a signaling pathway was first discovered, scientists thought they were very linear, okay, independent pathways, and there was no interconnection between them. Yeah, and then some proteins, we know now that they can participate in many pathways. And then depending on the cell type, depending on the signals that they're elucidating and they're participating in, and even when cells that are infected and damaged, or they've reached the end of their functional lifespan, you get very special signaling pathways. One of these is a whole program where the cell actually destroys itself. It is called cell suicide, controlled cell suicide, called apoptosis. Maybe you've heard of it before. Okay, it's a big complex system of signaling and of physical changes in the cell. What happens? Cellular agents digest DNA, they break down organelles, components within the cytoplasm. Ultimately, you start to break down this particular cell. Okay, how does it look like? The cell itself will begin to shrink. I don't want to say shrivel, but it'll start to develop little lobes, as you see here. You have a normal cell. This is a white blood cell. And this is another cell that is undergoing apoptosis. And you can see it has little bumps here, little lobes. They're called blebs or blebbing. And what's happening here, the cells, parts of the cells are becoming packaged up into vesicles. They're getting engulfed and they're getting digested by other cells, right? And so you have this effect that ultimately the cell's going to die, okay? And apoptosis is important because it can protect neighboring cells if the cell that is undergoing apoptosis is defective or has been infected or it is not working properly. The system activates to protect neighboring cells from any damage that could be done from this, defective cell, if I could say. And let's say leaking out contents of digestive enzymes that can damage other neighboring cells. Okay, so apoptosis plays an important role. It is not a bad thing. Definitely apoptosis is helpful for the body, and you can even have, trigger regeneration in some cases. It is important in embryonic development. We're going to take a look if I have the picture here. Okay, we're going to see this a little bit later with some visuals there. In embryonic development, where you have some initial signaling coming in from the outside, it will tell particular cells, okay, to start to break down very specific cells. And in terms of embryonic development, I'm going to show you it specifically. And interestingly, scientists have worked out this whole idea of apoptosis studying a very famous organism known as Cyanorhabditis elegans, commonly known as C. elegans, which you probably have heard of. Embryonic development of C. elegans, scientists have figured out what happens to each cell to the T, like to a specific number of cells. It's quite fascinating. An adult worm has only about 1,000 cells, okay? And it depends on the cell type, the animal type. So there's adult males, they have 1,333 cells. Adult hermaphrodites have 959 cells specifically. So scientists were able to figure out that cell suicide occurs 131 times during development of C. elegans, during at very specific points at a very particular cell lineages within this particular cell. And just to kind of read over this, this might be a little bit overkill for definitely for AP students, but for undergrads, discuss with your teacher if you need to know this in particular. What happens is that apoptosis, again, it's triggered by signals that activate a cascade of suicide proteins. That's pretty cool. And these proteins in cells that are destined to die, they will get activated. Okay, and there's two key apoptosis genes that is called CED3 and CED4. These are proteins that code for apoptosis, and they're usually present in cells, but they're in the inactive form. So activating them is that key thing in the signaling pathway. Okay, so regulation occurs at the level of protein activity rather than through gene activity and protein synthesis. Okay, so C. elegans, a protein in the outer mitochondrial membrane, a protein called CED9, right? Again, coming from the CED9 gene, it serves as a master regulator for apoptosis. It can act as a brake in the terms of whenever there's an absence of a, signal promoting molecule that induces apoptosis. Okay, so when a cell receives the death signal, the pathway will activate certain proteases and nucleases, and these are enzymes that can, break up proteins and DNA in the cell. That ultimately will induce cell death. Okay. Main proteases that are involved in apoptosis are referred to as caspases. Okay. One of these is CED3. Okay. Now, just to kind of show you what I was referring to. So in terms of no death signal, you have active CED9 protein. It will inhibit CED4. It will inhibit, so there's no caspase work occurring, so there's no digestion occurring. Okay? But if you get a ligand that is coming in to activate this death receptor, what happens? CED9 it becomes inactive, and then this will activate CED4 and 3 and this little cascade that will ultimately turn on the caspases, nucleases, and other proteases. Okay. Now again, this is not a bad thing. The cell does require this particular thing. Okay. So I want to talk about development in terms of, where do we see correct apoptosis with cell development. Sorry that I keep turning my head, guys, as I need to see if other individuals need the room. Okay. So again, as I mentioned before, apoptosis is necessary for vertebrate development. Okay. And maintenance. We do have many caspases, around 15 different caspases. Just a second, you guys. Okay. To carry out apoptosis, depending on the cell type, depending on the particular signal, you will get apoptosis starting. Okay. One major pathway does involve mitochondrial proteins. So the idea of breakdown, cell breakdown, it is with the use of lysosomes, but what actually starts the signaling mechanism could be triggered from mitochondrial proteins. Okay, and these mitochondrial proteins, they can form pores in the mitochondria that would ultimately leak the contents, release proteins, and leak proteins that will promote apoptosis. Okay. Interestingly, cytochrome C can, which works in, can work in the ETC, can also be involved in cell death process, right? So it could act as a cell death factor when released into the environment. Okay, and then mitochondrial apoptosis uses certain homologs or homologous proteins, right? So these are proteins that are very similar to CED3, CED4, and CED9. But again, it's involved with mitochondria. Okay. Ultimately, this will help relay a signal, induce relay proteins, okay, to promote this apoptotic signal. Okay. In terms of gateways into the apoptotic programs, again, as I mentioned before, relay proteins, they can relay the signal in itself. It can come from different sources. Ultimately, it will send a signal down. Okay. So I want to mention, two other types of alarm signals can originate from inside the cell. Okay, it could come from the nucleus when the DNA has physical damage, irreparable damage. You can elicit apoptosis, or when you have damage coming from the ER, where you have misfolding happening. Because if you have protein misfolding, you're going to have a deformed protein, which can lead to problems. Okay, so at the DNA level and at the protein folding level, you can have a problem. So, interestingly, mammalian cells can make life or death decisions to destroy itself. Okay, because you would rather, better to sacrifice yourself rather than create problems for the whole. Okay. And then lastly, I just want to talk about a built-in cell suicide mechanism. It's very important for development, okay, and maintenance in all mammals, and it has to do with the fingers. And many different things, but for humans, it can be with the fingers. Interestingly, of course, because it is seen in many animals, there is an evolutionary linkage. Okay, similarities of genes between nematodes and mammals that elucidate similar functions and processes. Okay, even you have the situation in fungi, right? So this can tell us that there is some signaling mechanism in early animal evolution, right? So we're all related in that sense, that we can have this apoptotic signaling pathway. It is important in vertebrates for normal development of the nervous system, immune, and even our appendages, right? Morphogenesis, hands and feet in humans, paws in other animals like mice. Very famous example, and you probably should know this as an example, is webbed feet. Okay? Webbed feet, when you have low apoptosis in developing limbs, webbed feet in ducks or other water birds, you tend to have this connection, and I'm going to show you guys. So let's say you have this particular developing hand in the mouse. You have the digits. Usually what happens is that you have in between the little digits here, you're having apoptosis occurring in these cells, ultimately that the tissue will die. And you have space in between the digits. Same thing happens to our fingers. Some individuals cannot, their body cannot correctly regulate apoptosis, and they tend to have more webbing, if I can say, that's a terrible term, a tissue that is connecting the fingers there. Okay, and the same thing happens with this type of waterfowl birds and other birds like chickens or land birds, that they do not have webbed feet. So it's just a regulation on the amount of apoptosis occurring. Okay, as I mentioned before, in humans, yes, you could have that same situation occurring. If you don't have appropriate apoptosis, then you will end up with webbed fingers and toes. Okay, And lastly, apoptosis has been seen to be involved in degenerative diseases of the nervous system, like Parkinson's and Alzheimer's disease. In some cases, cancer can result from failure to elucidate apoptosis, okay, to trigger apoptosis. Of course, you're going to have abnormal cells growing and growing and growing. Instead of dying out, this could be a big problem, okay? Like human melanoma are linked with faulty forms of the human version of CD4 protein. So very, very interesting. And these signaling pathways are very good areas for studying in cell biology and molecular biology. One of my favorite topics in biology. So with that being said, we have finished chapter 11. Just going to show you guys the chapter review for you to have. Zoom a little bit there. Okay, you can pause the video to help you collect the information there. And it's always good to try to answer these questions here, guys. Test your understanding. Okay. All right. Great. So thank you guys for watching. I hope this video was insightful for you to help you in your studies. Don't forget to like and subscribe, guys. It's always helpful for me continuing these videos. Okay. I will be producing more videos more often as we get into the summer period. All right. Thank you guys, and I'll see you in the next one.